In a metabolic pathway, the components are broken down into simpler forms wherein they release a lot of energy. So aerobic respiration takes place only in the presence of oxygen. So complex 4 is also called as cytochrome C oxidase complex. The energy that is released during the electron transport system is utilized in synthesizing ATP. Hello everyone, a warm welcome to another session on chapter 14, that is respiration in plants. I'm Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about an anaerobic process which is fermentation. So before that, in the first session, we had learnt about glycolysis. If you remember, in glycolysis, we had studied that the glucose molecule gets split into two molecules of pyruvate or pyruvic acid. So the fate of that pyruvic acid, whether it has to undergo fermentation or whether it has to enter into the next phase of aerobic pathway, which is Krebs cycle, which is also called as the citric acid cycle or the TCA, that will be decided based on the availability of the oxygen. For the pyruvic acid, if there is no availability of oxygen, then it will enter into the fermentation process, which we studied in the previous session of this chapter. So in fermentation, we had understood that the pyruvic acid can undergo fermentation in two forms. One is the lactic acid fermentation and the other one is the alcohol fermentation. So in the presence of microbes, if the pyruvic acid gets converted into ethanol, it is called as the alcoholic fermentation. And on the other hand, because of the unavailability of oxygen, if pyruvic acid gets converted into lactic acid, then it is called as lactic acid fermentation. So if this pyruvic acid gets enough of oxygen, then it will enter into the aerobic pathway, which is called as the Krebs cycle. The cycle is named based on the findings done by a scientist, that is Hans Krebs. And also, it is also called as the citric acid cycle, and it is also called as the tricarboxylic acid cycle. So, before entering into the Krebs cycle, this particular pyruvic acid, they undergo decarboxylation. That is, the carbon is lost in the form of carbon dioxide, wherein pyruvic acid gets converted into acetyl-CoA. That acetyl-CoA gets converted into oxaloacetic acid, and it continues with the Krebs cycle. So for all this process to occur, a lot of energy is needed, and we all know that energy is supplied by the mitochondria of the cell, which is called as the powerhouse of the cell. And energy, it is in the form of ATP, right? So to do our daily activities, we need a lot of energy. That energy is got from the food we eat. But that food should be broken down into the form of energy, right? That is, for example, say glucose. It should be broken down into the form of energy. So if it has to be broken down into energy, a lot of electrons are also needed, which should be carried through different carriers, so that at the end or at the terminal stage of the movement of the electrons, the ATPs can be generated. So that particular pathway wherein a lot of energy is being generated by the traveling of the electrons is called as the electron transport system. So in today's session, we shall deal with electron transport system, and also we shall understand what is oxidative phosphorylation. So in the previous chapter of photosynthesis, we had studied photophosphorylation, right? But that is in the presence of sunlight. But when the plants don't make use of the sunlight, say for example the mitochondria, when it doesn't make use of the sunlight at that particular process, or if the plants need to make use of the sunlight, they need chloroplast, right? But remember, in the first session of this chapter, I had told you that not all the cells of the plants possess chloroplast. So those cells of the plants which do not possess chloroplast, they actually go in for the respiration process. And if chloroplasts are there, they go in for the photosynthesis process. So therefore here, the phosphorylation or the addition of the phosphate group to the ADPs in order to generate energy in the form of ATPs is by oxidative phosphorylation. So all this in detail we shall study in today's session. So to begin with the chapter, we shall first see what is the electron transport system, and also I've put the heading here as electron transport system and oxidative phosphorylation, because both go hand in hand, because it is at the end of this process that is there. At the terminal stage of the process, oxidative phosphorylation will occur. So that is why both go hand in hand. So that is why we put both of the topics are put together, that is electron transport system and oxidative phosphorylation. So let's see what is this electron transport system. So it is a metabolic pathway. We all know that in a metabolic pathway, the components are broken down into simpler forms wherein they release a lot of energy, right? So that's why here also energy is being released or energy is being made by the splitting up of the compounds. So therefore, it is a metabolic pathway through which electron passes. As the name itself says, electron transport system. So here, the electrons are carried from one complex to another complex. So here, the electron passes from one carrier to another, and this is called as the electron transport system. So if you see this pathway here, you will understand. So there are four types of complexes, which is called as complex 1, complex 2, then we have complex 3, then comes complex 4. And you should always remember the name of the complex, that is complex 1, 2, 3 and 4 are always written in the Roman numerals. So it should be written in the Roman numerals. So here, if you see, through these different complexes, the electrons are being carried or they are moving, and it results in a chain of movement of electrons or a system of movement of electrons. So therefore, this pathway is called as the electron transport system. And it is present in the inner mitochondrial membrane. So if you look at the structure of the mitochondria, we know that the mitochondria is covered by a membrane wherein it has a outer membrane, inner membrane, and in between the outer and the inner membrane there is a space which is called as the intermembrane space, and within the inner membrane is present the fluid matrix. So for your easy understanding, if you just draw the diagram of a mitochondria, we can find the outer membrane, which is outer mitochondrial membrane or outer membrane. Then inner to it is present the inner membrane or the inner mitochondrial membrane. Inner membrane, and then in between the outer membrane and the inner membrane there is a space that is present. So that space is called as the intermembrane space. So outer membrane, inner membrane, in between the outer membrane and the inner membrane there is a space which is called as inter. So intermembrane space. And inner to the inner membrane is present the matrix. So it is the matrix present inner to the inner membrane. So we should always know that in the matrix, actually, a aerobic pathway takes place in the matrix, which is called as the Krebs cycle. So in the matrix, usually the Krebs cycle will occur. So where does this electron transport system occurs? The electron transport system actually occurs in the inner membrane. So it is in this particular inner membrane region, the electron transport system will occur. So for this electron transport system, electrons are needed. From where will it get the electrons? So the electrons are got from the Krebs cycle. So in the Krebs cycle, the NADH, so we know that in the Krebs cycle, NADH and FADH2s are produced, right? So the NADH and the FADH2, that are formed in the Krebs cycle, they actually supply the electrons for the electron transport system, and those electrons actually travel through different carriers or complexes. So that is how it gets its electrons. So electron transport system, it occurs in the inner mitochondrial membrane, and electrons from NADH, so electrons that are produced from NADH during the process of Krebs cycle, it is produced in the mitochondrial matrix. So why is it produced in the mitochondrial matrix? Because the Krebs cycle occurs in the mitochondrial matrix, or you can also write it as it is produced in the citric acid cycle, or it is produced in the tricarboxylic acid cycle. All the names are same because citric acid cycle is also called as the Krebs cycle. So it is produced in the, and where does this Krebs cycle or the citric acid cycle occur? It occurs in the matrix of the mitochondria. So therefore, electrons that are produced by the NADH and the FADH2 that are formed during the citric acid cycle or the Krebs cycle in the matrix of the mitochondria, they will be supplied to the electron transport system. So they will be oxidized by a NADH dehydrogenase complex. So now I need to make you understand. So here, if you look at this electron transport system, it looks very complicated, but once you understand the concept, it is actually very, very easy. So the same thing which I drew here, that the same thing is being represented here. So if you look here, you can see the inner mitochondrial membrane. So imagine this is the mitochondria, which I'm drawing a huge outer boundary here. It is the mitochondria. So in the mitochondria, what is there? This is the outer membrane. Outer mitochondrial membrane. So this is the outer membrane. Then inner to the outer membrane. After the outer membrane, what is there? The inner membrane is there, right? So we have the inner mitochondrial membrane, if you can see. And remember, I had told you between the inner membrane. So here also the inner membrane, I'll just draw a diagram like this. Again, one circle, which is the inner membrane of the mitochondria. Yeah. So this is the inner membrane of the mitochondria. Inner membrane. So here, this is the inner membrane. So this one is the inner membrane. So between the outer membrane and the inner membrane, there is an intermembrane space. If you see, so that is the intermembrane space that is present between the outer membrane and the inner membrane. So here, and within the inner membrane is present the matrix. It is just this one which is being shown in an expanded form without any boundaries here. That's it. So this is the matrix that is present. So in the matrix, NADH and H plus, all that is being there. Why? Because NADH and all that would have been produced during the Krebs cycle or the citric acid cycle, right? So here, this NADH, it undergoes dehydrogen, means it is losing a hydrogen, right? So here what happens? It is losing a hydrogen. So when it loses the hydrogen, it will remain as NAD. It gets converted into NAD, and that hydrogen that is being lost in the matrix, it will pass through the inner mitochondrial membrane through this complex, and it will enter into the intermembrane space. Now, from where did it travel? The hydrogen traveled from a region of higher concentration, that is in the matrix, to a region of the lower concentration, that is intermembrane space. So before understanding that, you should know about all the different complexes, that is complex 1, complex 2, complex 3 and complex 4. So they have different names. So if you study them along with the names, it is easy for us to even more understand. So first I'm talking about the complex 1 in the electron transport system. So this particular complex 1 here in the matrix, the NADH that is produced during the Krebs cycle, they undergo dehydrogenation. That is, they lose their hydrogen. So why do they lose their hydrogen? Because of the presence of NADH dehydrogenase. So NADH dehydrogenase, so the complex 1 is also, so this is complex 1. If you can see here, this is complex 1. So the complex 1 is also called as NADH dehydrogenase complex. So why is it called NADH dehydrogenase? Why? Because from the NADH that is present in the matrix of the mitochondria, the hydrogen is being removed. So that is why it is called as NADH dehydrogenase. So why is it called a complex? It is called a complex because here flavin mononucleotide. So FMN, I'll just write it here. FMN is called as flavin mononucleotide. Mononucleotide. So FMN and FES. So FES is nothing but ferrous sulfate. Ferrous sulfate or iron sulfate, also we can call it as iron sulfate. So here the flavin mononucleotide and iron sulfate are present in the form of a complex. They actually are present in this particular complex. So that is why it is called as a complex. So that is why complex 1 is also called as NADH dehydrogenase complex. So the electrons, as I told you, as the hydrogen is being lost here, that hydrogen from the matrix, it will move into the intermembrane space, passing the inner mitochondrial membrane. So there is something which should carry the hydrogen. So what is that? It is the NADH dehydrogenase complex that actually helps in the movement of the hydrogen through the inner mitochondrial membrane. And apart from that, this particular complex, which is made up of ferrous mononucleotide, which is made up of flavin mononucleotide and ferrous sulfate or iron sulfate, they help in the passing or movement of electrons. So they help in the movement of the electrons, and these electrons will be taken by the ubiquitin, and that ubiquitin will carry the electrons into the cytochrome, that is, or cytochrome BC1 complex or complex 3. And again here also one more thing you should remember. Complex 2 also supplies the electrons. If you can see an arrow mark which is directed towards the cytochrome BC1 complex, which is the complex 3. So here the main function of the complex 3 is to receive the electron from complex 1 as well as from complex 2, if you notice here. So here electrons will be carried. Okay. So now the electrons are taken in by ubiquitin. So UQ here, it is nothing but ubiquitin. So that will be taken, the electron that is there, it will be taken by the ubiquitin, and that ubiquitin will transfer the electron to the complex 3. Next we have complex 2 here. So complex 2, it is named as succinate dehydrogenase complex. If you can see here, it is named as succinate dehydrogenase complex. So again here dehydrogenase, why the word dehydrogenase here? Because hydrogen is being removed. From where is the hydrogen being removed? The hydrogen is being removed from succinate. So when succinate gets converted into fumarate, at that time hydrogen is being released, and that hydrogen will also move into the intermembrane space. That is why it is called a succinate dehydrogenase. Again, why is it called a complex? It is called a complex because here FAD and iron sulfide is being produced, and not just that, FADs, from FADs also, that is FADH2 also, the hydrogen is being released. So you should remember every NADH or FADH, they will give 2 hydrogen. So here also 4H plus, here also 4H plus. So here they give the hydrogen molecules into the intermembrane space, and not just that, here also they will give in the electrons. So these electrons will be taken in by the ubiquitin and they will enter into the, so that is complex 3. So now in the complex 3, the complex 3 is called as cytochrome BC1 complex. So why is it called as cytochrome BC1 complex? Because it is made up of different types of cytochromes, such as cytochrome C1 and cytochrome B, and also cytochrome C. So here Apart from that, it has a protein which is nothing but made up of heme, and therefore it also has iron sulfate. So that is why it is called as a complex. So all these also help in the movement of the electrons. So the electrons travel through the cytochrome C complex. So don't get confused. The function of the complex 3, which is called as cytochrome Bc1 complex, is to receive the electrons from the complex 1 and the complex 2. So those electrons which are received from the complex 1 and complex 2 by the complex 3, it will be carried by the cytochrome C into the complex 4. So if you can see here, to the complex 4, it will be carried to complex 4. So complex 4 is also called as cytochrome C oxidase complex. So this is the last stage. So why is it called as oxidase? So it is called as oxidase because here oxidation or addition of oxygen molecule will occur here. So that is why. So here if you can see, cytochrome C oxidase is made up of cytochrome C. And also apart from that, it is made up of different types of cytochrome such as cytochrome A and cytochrome A3, and also it is made up of copper, that is copper A and copper B. So all these complexes, it also helps in the carrying of the electrons. Now here what happens? Here oxidation is occurring wherein to the H plus ions, half a molecule of oxygen is being added, resulting in the formation of water, and this H plus, that 2H plus that is formed, will also travel into the intermembrane space. Now in the intermembrane space, lots of hydrogen ion concentration has increased, resulting in a gradient, that is in a proton gradient. So this concentration gradient, what will happen if the concentration gradient increases? So in concentration gradient increases means because here the positive charges have increased more. So if you can see here, why? Because all the H plus has traveled from the matrix of the mitochondria into the intermembrane space. So here the positive charges have increased more in the intermembrane space, whereas in the matrix the negative charge has increased more, and that positive charge which has increased more in the intermembrane space because of the hydrogen ion, because of the concentration of the hydrogen ion, a concentration gradient is being created, and that concentration gradient will not allow the hydrogen ions to pass through the inner mitochondrial membrane. Why? Because the concentration gradient will make the electrochemical, or in other words I can say a electrochemical gradient is being created. That electrochemical gradient will make the inner mitochondrial membrane impermeable to the H plus ions. Therefore, the H plus ions cannot go back into the mitochondrial matrix. So, it is during that time certain proteins or certain complexes come to help. So, we shall see about that in the next slide now. In this particular slide, we have understood how electrons are transported through different complexes, then how the hydrogen ions travel from the matrix to the intermembrane space, and in the intermembrane space, when the concentration of hydrogen ion increased, it resulted in the creation of a electrochemical gradient, and that electrochemical gradient is now making the inner mitochondrial membrane impermeable to the hydrogen ions, therefore not letting the hydrogen ions to pass into the mitochondrial matrix. Now, how will the hydrogen ions pass back to the mitochondrial matrix? That is the question. So, we will look into it in the next slide. So, here I was talking about complex 4. So, the complex 4 refers to cytochrome C oxidase. So, again, we'll talk about it again. So it is a complex because it contains cytochrome A and cytochrome A3, and also it is made up of two copper centers which are called as copper A and copper B. So all these are present in that, and when the electrons pass from one carrier to another via the complex 1 to 4, that is in the electron transport chain, when the electrons passes through different complexes, that is right from complex 1 to complex 4, here they are coupled to ATP synthase in the complex 5. So whatever electrons travel from complex 1 to complex 4 in the complex 5, so this particular we can name it as complex 5. So in the complex 5, they all pool into the ATP synthase enzyme. So that is the time when ATP synthase enzyme actually helps in the addition of a phosphate group to the ADP, and therefore phosphorylation will occur, and that is when ADP becomes ATP. So that is what occurs in the complex 5. So here what happens? Again, to make you understand, so electrons have passed through different carriers, right? They have passed from complex 1, then from complex 1 and complex 2, all the electrons entered into the complex 3. In the complex 3, through the cytochrome C, the electrons was carried to the complex 4, and from the complex 4, whatever electrons are there, they will enter into the complex 5. So this particular region is the complex 5. So in this complex 5, all the electrons will move towards, or it will be accepted by an enzyme called as ATP synthase. So that ATP synthase enzyme, it will collect the electrons and it will help in the production of ATP from ADP and inorganic phosphate. So to ADP, one more phosphate group gets added. What will become? It will become an ATP, that is a diphosphate will become a triphosphate, right? So it will get converted into an ATP. So the number of molecules actually, that is the number of ATP. So now the question is, okay, how many ATPs will get synthesized? So how many ATPs should be synthesized by this particular oxidative phosphorylation will actually depend on the following features, that is the nature of the electron donor. So what is the nature of NADH, all that, or the nature of the electron donor? Based on that, the number of ATPs that are synthesized at the terminal stage of the electron transport system will be decided. And not just that, oxidation of one molecule of NADH will give rise to 3 molecules of ATP. So remember, one molecule of NADH, when it gets oxidized, it will give rise to 3 molecules of ATP, while that one molecule of FADH2 will produce 2 molecules of ATP. So what did I tell you? The ATP that is produced at the end of the electron transport chain depends on the electron donor that is participating. So what are the electron donors that are participating in this particular electron transport system here? It is the NADH and the FADH2, right? Because they are the ones who are donating the electrons. So if NADH is the one that is donating the electrons, then for one molecule of NADH, 3 molecules of ATP will be produced at the end of the electron transport system. And on the other hand, if FADH is the source, that is the one that is giving the electrons for the electron transport system to take place, then at the end of the electron transport system, 2 molecules of ATP will be produced. So for you to easily understand, NADH will produce 3 molecules of ATP and FADH2 will produce 2 molecules of ATP. So the electron donor, that is NADH and FADH2, are the ones that actually decide how much ATP should be produced at the end of this particular electron transport reaction. So far we had not talked about FADH2, we had just talked about NADH. So since NADH is participating here, so therefore 3 molecules of ATP will be produced at the end of this particular reaction. So now we know about the electron transport system, and we also know about the complex 5, wherein the electrons will go and pool into the ATP synthase, and that ATP synthase will help in the addition of a phosphate group to the adenosine diphosphate, therefore forming a adenosine triphosphate, which is nothing but ATP, and that ATP is nothing but the energy that is utilized by all the living organisms for various biochemical pathways or for our daily activities and all that. So now let us concentrate on the complex 5. What exactly is happening in the complex 5? Because if you remember, I had told you the H plus ions cannot come back into the mitochondrial matrix, but actually the H plus ions can come into the mitochondrial matrix back again. How is it so? We shall see in the next step, that is oxidative phosphorylation. So here we had studied that aerobic process of respiration, it takes place only in the presence of oxygen. That is why it is called as aerobic, right? Here, oxidation will occur. That is, the addition of oxygen molecules will occur. So, aerobic respiration takes place only in the presence of oxygen. But what we have seen in the electron transport system is we saw that only in the last stage of the electron transport system, half a molecule of oxygen got added, resulting in the production of water, right? So only in the terminal end, that is, we thought that the role of oxygen was limited only to the terminal stage of the electron transport system, as we saw earlier. But we should always remember that for the movement of electrons, that is for the removal of hydrogen or for the movement of electrons, especially for the removal of hydrogen, that is from NADH, H+, if the hydrogen has to be removed, then oxygen is very needed. So oxygen is needed not just in the terminal stage of the electron transport system, but or at the end stage of the electron transport system, but it is also needed, or oxygen is very vital, or it is very important for removing the hydrogen from the NADH in the matrix of the mitochondria. Also oxygen is very important. So throughout the electron transport system, oxygen is very important. And here, oxygen, not just that, it acts as a hydrogen acceptor, right? So we had seen here, if you see here, they act as a hydrogen acceptor. So we have seen in the previous slide. So oxygen acts as the final hydrogen acceptor. Presence of the oxygen molecule here, they try to accept the hydrogen here. So they act as the oxygen acts as the final hydrogen acceptor, and unlike photophosphorylation. So photophosphorylation occurs during photosynthesis. So in photophosphorylation, what happens? Light, functions like a source of energy. That is, the light energy is utilized for the production of here a proton gradient. So light energy is utilized for the production of a proton gradient, which is required for phosphorylation. So we had studied about all that in the previous cycle, right? That is, in the previous slide we had studied, wherein, say for example, during photosynthesis, what happens is the mitochondria makes use, or the chloroplasts, they make use of the sunlight energy, and these actually helps in addition of a phosphate group into the ADP, therefore resulting in phosphorylation. And because light is involved there, it is called as photophosphorylation. But in the case of electron transport system, here light is not being involved. So when light is not involved, how will phosphorylation occur? So here phosphorylation will occur by the addition of an oxygen molecule, by the presence of an oxygen molecule. So oxygen molecule it helps in the addition of a phosphate group into the ADP. So that is why it is called as oxidative phosphorylation. So here in respiration, so now we are talking about respiration. So there in photosynthesis, photophosphorylation will occur. But in respiration, oxidative phosphorylation will occur. So in respiration, the energy of oxidation reduction reaction is utilized for the same process, that is for phosphorylation process. So because of oxidative reduction reaction, the phosphate molecule will be added to adenosine diphosphate. Therefore, it will get converted into adenine or adenosine triphosphate. And it is for this reason why here, because oxygen is the one that actually, or because both oxidation and reduction reactions are involved in the phosphorylation process, this particular step, it is called as oxidative phosphorylation. So here actually in the complex 5, oxidative phosphorylation will occur. Why? Because here phosphorylation takes place by oxidative reduction reaction. Hydrogen will get lost, oxygen will get added. So that is how it occurs. So this is about, now we understood what is oxidative phosphorylation. So this is very important for the last step to understand this. So that is why oxidative phosphorylation occurs in the terminal stage of the electron transport system. So here what happens during oxidative phosphorylation? So we know that a lot of energy will be released during the electron transport system during the movement of the electron. So that energy that is released, it will be utilized for synthesizing ATP. So how will ATP be synthesized? In the presence of ATP synthase. So what did I tell you? Electrons will travel through complex 1, the electrons will travel through complex 2, and those electrons will pool into complex 3. From complex 3 they will be carried into complex 4, and from complex 4 they will move into the complex 5 wherein ATP synthase enzyme is being produced. That ATP synthase enzyme receives all the electrons, therefore it will get enough energy to allow the formation of ATPs, right? So here that is why ATP synthase helps in the formation of the ATPs, which takes place in complex 5. And this complex consists of two major components, F1 and F0. So in this particular slide we had seen this is complex 5. Here it is made up of two important components, F0 and F1 component. The same thing they have shown it here in a bigger manner. So here the F0 complex is present, F1 complex is present, and which is nothing but the complex 5. It is the complex 5, right? So here the F0, this complex, it consists of two major components, that is F1 and F0. So the F1 headpiece that is there, it is a peripheral membrane protein. So this F1, it is a peripheral membrane protein. So this peripheral membrane protein that is there, which is nothing but F1, and this is the one that actually contains the site for the synthesis of ATP from ADP and inorganic phosphate. So that is how here ATP, so to this ADP, that is adenosine diphosphate, a phosphate group will be added. How? In the presence of the ATP synthase enzyme that is synthesized by the F1 complex, which is a peripheral membrane protein, and therefore it will get converted into ATP. So here what happens? 3 molecules of ATPs will be produced if it is a NADH that is taking part in the electron transport system. On the other hand, if it is a FADH that is taking part in the electron transport system, 2 molecules of ATP will be produced. So this is about the oxidative phosphorylation, which we have understood. So here, so F0, so we talked about F1. So F0 that is there, it is an integral membrane protein complex and it forms a channel through which the protons cross the inner membrane. So if you can see here F0 actually helps the protons to cross the inner membrane of the mitochondria. So in the intermembrane space of the mitochondria, a lot of proton concentration was there, which resulted in an electrochemical gradient, therefore not allowing the protons to pass through the inner membrane of the mitochondria and then to enter into the matrix. But here in the complex 5, there is a complex called as F0 complex, which is a intermembrane protein complex. That protein complex actually helps the protons to move from the inner mitochondrial membrane back into the matrix. So it will move from there into this. So that is how, so far in electron transport system we had not completely discussed about the last stage. Why? Because I was waiting for oxidative phosphorylation. So at that time we can discuss about it. So if I show you here in the previous picture itself, you can see here F0, it actually helps in the movement of the electrons through the inner membrane, and that electron will enter into the matrix back. So that electron actually moves here. And that is how one more electron gets added there. So I told you 2H, 1H here, 2H plus will move. This Again 2H plus will move, therefore 4H plus. So that is how it gets added. The same thing is shown here as well. So here they help in the passage of protons across the inner membrane. So the passage of protons to this channel, so this particular channel that is there wherein the proton passes, it is actually coupled to the catalytic site of the F1 component for the production of ATP. So this also helps in the production of ATP. F0 is always coupled with F1. Both of these help in the passage of the electrons and passage of the protons, and also they help in the production of a lot of energy in the form of ATP. And for each ATP produced, actually 2H plus passes. So remember, if ATP has to be produced, then the passage of 2H plus from the intermembrane space into the matrix of the mitochondria is also very, very important. So 2H plus passes through F0 complex from the intermembrane space to the matrix down the electrochemical proton gradient, which we had talked about it earlier. So what happens here? So I'll just explain it in this particular diagram itself. So the electrons have reached into the complex 4. From the complex 4, they have all pooled into complex Y. Why they have pooled into complex Y? Because they need to provide energy for the ATP synthase in order to undergo oxidative phosphorylation, right? So wherein the phosphate groups get added into the ADPs, therefore the ATP can be produced. Apart from that, this is made up of two important complexes, that is F0 complex and F1 complex. The F0 complex actually helps in the movement of proton from the intermembrane space. It will help it to pass through the inner mitochondrial membrane and then into the matrix. So here, every time 2H plus protons, two molecules of hydrogen will pass through this. So these two molecules of hydrogen will also be utilized for the production of ATP. So they are also needed if ATP has to be produced. And the F1 actually also helps in a particular process wherein the F1 also helps in the formation of the ATP. So all this, so they help in breaking of the proton gradient, that electrochemical gradient that is there. So therefore the H plus ions can pass through the membrane and enter back into the matrix. So that is how they help. So this was about electron transport system and oxidative phosphorylation, which we studied. If we understand the concept very well, it is easy for us to understand and remember it. So therefore, this was about electron transport system and oxidative phosphorylation. So I hope you understood this session. We'll meet again in the coming session. So in the coming session, we shall discuss about the respiratory balance sheet. So far we had understood a lot of ATPs are being produced, right? Both in the aerobic respiration, that is during the aerobic respiration, when it undergoes glycolysis or when it undergoes decarboxylation and when it undergoes a Krebs cycle, how much amount of ATP is generated. To calculate that, we need to put a respiratory balance sheet. So what is respiratory balance sheets? All that we shall study in the coming session, and we shall also discuss an important pathway called as the amphibolic pathway. So we shall meet again in the coming session. Thank you.